Introduction. Glutamine, an amino acid often referred to as the conditionally essential amino acid, plays a vital role in numerous biochemical pathways within the human body. From its involvement in nitrogen metabolism to its contribution to glutathione synthesis, glutamine serves as a key player in various physiological processes. This script aims to provide a comprehensive overview of glutamine metabolism, its synthesis and utilization, transporters responsible for cellular uptake, enzymatic reactions involving glutaminase, and its implications in several areas such as cancer metabolism, gut health, exercise physiology, neurological disorders, health span/longevity, and critical illness/trauma. By delving into the intricacies of glutamine's biochemical functions, we hope to enhance our understanding of its significance in human health and disease. So, let's explore the fascinating world of glutamine metabolism. Glutamine, a non-essential amino acid, is synthesized through a series of enzymatic reactions predominantly within skeletal muscle and the liver. The first step in glutamine biosynthesis involves the conversion of glutamate, an essential intermediate, to glutamine via the enzyme glutamine synthetase. Glutamine synthetase condenses glutamate with ammonia, derived from the amino acids alanine and histidine, to form glutamine. Subsequently, glutamine can be utilized for various metabolic processes throughout the body. In addition to the endogenous synthesis, glutamine can also be obtained from the diet, particularly in protein-rich foods such as meat, eggs, and dairy products. Upon ingestion, dietary glutamine undergoes digestion and is absorbed into the bloodstream through the intestines, where it becomes available for uptake and utilization by various target tissues. The synthesis of glutamine occurs predominantly in skeletal muscle and the liver, which possess high levels of glutamine synthetase. Skeletal muscle is responsible for the synthesis of approximately 90% of the total glutamine produced in the body, while the liver contributes to the remaining 10%. This distribution ensures an ample supply of glutamine to meet the demands of different tissues. Once synthesized or ingested, glutamine serves as a primary nitrogen carrier. It donates nitrogen for the synthesis of nucleotides, non-essential amino acids, and other nitrogen-containing compounds essential for cellular function and growth. Glutamine also acts as a precursor for the synthesis of glutathione, an important antioxidant molecule involved in cellular defense against oxidative stress. Furthermore, glutamine plays a critical role in maintaining the acid-base balance within the body. It acts as a buffer, accepting protons released during metabolic reactions and contributing to the regulation of pH. This ability of glutamine to regulate acid-base balance is particularly important in organs with high metabolic demands, such as the kidneys. To fulfill its role as a vital nutrient, glutamine must be transported across cell membranes for utilization by various tissues. The cellular uptake of glutamine is predominantly mediated by specific glutamine transporters, namely the sodium-dependent neutral amino acid transporters, SNATs, and the alanine-serine cysteine transporter 2, ASCT2. SNATs, encoded by the SLC38 gene family, are responsible for the majority of glutamine uptake in most tissues. These transporters utilize the sodium gradient established by the sodium-potassium ATPase to transport glutamine into the cell. ASCT2, on the other hand, primarily facilitates glutamine uptake in the brain and certain cancer cells. The expression and activity of glutamine transporters are tightly regulated in response to various physiological and pathological conditions, ensuring an appropriate availability of glutamine for different tissues. Glutaminase, an enzyme involved in glutamine metabolism, catalyzes the hydrolysis of glutamine to yield glutamate and ammonia. This enzymatic reaction plays a crucial role in glutamine utilization by various tissues such as the kidneys, intestines, and brain. There are two isoforms of glutaminase, namely kidney-type glutaminase, KGA, and liver-type glutaminase, LGA. KGA is predominantly expressed in the kidneys and intestines, while LGA is primarily found in the liver. Both isoforms contribute to cellular glutamine catabolism and subsequent utilization of the released glutamate. The produced glutamate from glutaminase activity can be further metabolized through the citric acid cycle to generate energy or serve as a precursor for the synthesis of other amino acids, neurotransmitters, and biomolecules essential for cell function. As an essential component of nitrogen metabolism, glutamine plays a pivotal role in facilitating the transport and balance of nitrogen within the body. Through the process of transamination, glutamine donates its amine nitrogen to alpha-keto acids, forming amino acids that are vital for protein synthesis. Moreover, glutamine acts as a nitrogen shuttle between tissues during times of increased demand. For instance, during periods of fasting or stress, skeletal muscle releases glutamine, which is subsequently taken up by the liver and kidney to furnish the necessary nitrogen for the synthesis of glucose, urea, and other nitrogenous compounds. In addition, Glutamine metabolism also regulates the levels of ammonia, a toxic byproduct of nitrogen metabolism. By converting glutamine to glutamate and ammonia through the action of glutaminase, excess ammonia can be removed from the body via the urea cycle, averting potential toxicity. Glutathione, a potent endogenous antioxidant, plays a crucial role in cellular defense against oxidative stress. Glutamine serves as a critical precursor for glutathione synthesis, contributing to the maintenance of cellular redox homeostasis. Within cells, glutamine is converted to glutamate via glutaminase. Glutamate, in turn, reacts with cysteine and glycine, catalyzed by glutathione synthetase, to form glutathione. Glutathione's thiol group provides a potent reducing power, enabling it to scavenge reactive oxygen species, ROS, and protect cells from oxidative damage. The ability of glutamine to replenish glutathione levels makes it crucial in maintaining cellular integrity and protecting against oxidative stress-related diseases. Cancer cells exhibit altered metabolic pathways to support their rapid growth and proliferation. Glutamine metabolism plays a prominent role in fueling tumor growth by providing a carbon and nitrogen source for biosynthesis. In cancer cells, glutamine is avidly taken up via the upregulated expression of glutamine transporters, enabling increased glutamine utilization. Once inside the tumor cells, glutamine is converted to glutamate by glutaminase, contributing to the production of ATP, an essential energy source for cancer cell proliferation. Moreover, glutamine metabolism fuels the synthesis of other critical biomolecules necessary for tumor growth, such as nucleotides and amino acids. This dependency on glutamine makes it an attractive target for the development of novel anti-cancer therapies. The gastrointestinal tract serves as a primary site of glutamine utilization, particularly in supporting the integrity and function of the gut mucosa. Glutamine is crucial for the maintenance of intestinal barrier function, immune response modulation, and prevention of bacterial translocation. Within the intestines, glutamine is consumed by the rapidly dividing epithelial cells lining the gut. It acts as a fuel source and supports the synthesis of proteins essential for cellular growth and proliferation. Additionally, glutamine plays a role in regulating gut immune function by influencing the production of cytokines and modulating immune cell activity. The provision of supplemental glutamine has been investigated as a therapeutic strategy for various gastrointestinal disorders, including inflammatory bowel disease and intestinal injury. During periods of intense exercise or endurance training, the demand for energy and nitrogenous compounds increases significantly. Glutamine, being a key nitrogen carrier and energy substrate, plays a vital role in mediating exercise-induced metabolic adaptations. Exercise stimulates the release of glutamine from skeletal muscle, which is then taken up by other tissues such as the liver and intestines, contributing to the synthesis of glucose, amino acids, and other metabolites necessary for sustaining energy levels. Moreover, glutamine supplementation has been suggested to enhance immune function, reduce exercise-induced muscle damage, and improve recovery following strenuous physical activity. Emerging evidence indicates the involvement of glutamine metabolism in the pathogenesis of various neurological disorders, including Alzheimer's disease, epilepsy, and amyotrophic lateral sclerosis, ALS. Glutamine acts as a precursor for neurotransmitters such as glutamate and gamma-aminobutyric acid, GABA, which play essential roles in neuronal communication and excitatory-inhibitory balance. Dysregulation of glutamine metabolism can disrupt neurotransmitter homeostasis, leading to neuronal dysfunction and disease progression. Furthermore, glutamine's role in protecting against oxidative stress and modulating inflammation makes it an attractive target for potential therapeutic interventions in neurological disorders. The aging process is associated with various metabolic changes and an increased susceptibility to age-related diseases. Glutamine metabolism has garnered attention as a potential regulator of health span and longevity due to its involvement in key cellular processes such as energy metabolism, DNA/RNA synthesis, and antioxidant defense. Research suggests that dietary interventions that modulate glutamine availability and metabolism may have implications for extending health span and promoting healthy aging. However, further studies are needed to fully elucidate the underlying mechanisms and assess the effectiveness of such interventions. During critical illness and trauma, the body undergoes significant metabolic stress and metabolic derangements. Glutamine, being a vital nutrient for immune cells and rapidly dividing tissues, possesses the potential to influence outcomes in critically ill patients. The metabolic demands of critical illness often exceed the body's ability to produce sufficient glutamine, leading to glutamine depletion and impaired immune function. Glutamine supplementation has been investigated as a potential therapeutic strategy to mitigate these effects, supporting immune function, enhancing gut integrity, and improving patient outcomes. Overall, Optimizing glutamine availability and metabolism in critically ill and traumatized patients holds promise as a therapeutic approach to improve recovery and reduce the risk of complications. To summarize, glutamine is an essential player in human physiological processes, impacting various aspects such as nitrogen balance, glutathione metabolism, and cellular uptake. It has been found to have implications in cancer, gut health, exercise, neurological disorders, health span, and critical illness. By studying the intricate details of glutamine metabolism, we can potentially develop new treatment strategies and improve patient outcomes. Continued research and clinical studies will enhance our knowledge and appreciation of this multifaceted amino acid and its role in human health.